Hello, everyone. Welcome to today's Field Advisor webinar. My name is Kelsey Litchfield. I'm the Agronomic Outreach Manager and Editor of Field Advisor, and I'm being joined by Lauren Hill, who's also running tech behind the scenes today. If you're new and have never attended a Field Advisor webinar, welcome. If you're returning, welcome again. If you haven't heard of Field Advisor before, we are funded by the Illinois Soybean Checkoff program, and Field Advisor provides the latest agronomic information and on-farm research from local experts to help Illinois soybean producers improve profitability and yield. We're an independent, unbiased media platforms. We have blogs, videos, a podcast, and of course the webinar that you're watching today. Before we begin, some housekeeping items. This webinar is being recorded after the live session, and then we'll put it on Field Advisor's YouTube channel for you to watch back. And a survey will also be emailed out. So once that survey comes out, it'll also contain the link to that recording as well. CEU credits, one is available in pest management. If you are watching the webinar live, a QR code will be available after the presentation. And if you're watching the recording of this webinar, we recommend self-reporting for the CEU credit. If you email fieldadvisor@illsoy.org, that's fieldadvisor@illsoy.org, we can help you out to self-report. Of course, questions are welcome. Thank you for those that sent in questions ahead of time. We will share those with Dr. Tranel at the end. But if you have questions throughout the webinar, you're more than welcome to put it in the Q&A box. Now, let me introduce our presenter today, Dr. Patrick Tranel. He joined the University of Illinois in the late 1990s and has been teaching undergrad and graduate students about weed science both on campus and more recently through the online Master of Science program. As weeds have evolved over the years, so has the research Dr. Tranel conducts. He uses a molecular and genomic approaches to better understand how weeds evolved to the various tools farmers use to control them. He is a native of Galena, Illinois, and he earned his Bachelor of Science in Agronomy from Iowa State University, Master's in Weed Science from Washington State, and a doctorate in botany from Michigan State University. Dr. Tranel, the floor is now yours.
Thank you, Kelsey, for that introduction. Let me pull up my screen here. Okay, yes, thank you again, Kelsey, for the introduction. Thank you for this opportunity on your media platform here to share some of my research, some of my thoughts about glufosinate and utilizing glufosinate for water hemp control. So here's an outline of my presentation today. I'm going to start out with introduction to water hemp, which I'm sure 99% of the audience is all too familiar with already, but just to bring everyone up to speed, talk a little bit about this weed and the current challenges we're facing with herbicide resistance in it. And then I'll move on to glufosinate, talk a little bit, introduce that herbicide and where we stand with resistance to glufosinate. And then I'll move into some recent research that we've been involved with, funded by ISA, really to look at variable glyphosate or glufosinate responses in water hemp, looking at different factors as well as population differences that might be contributing to this variable or inconsistent performance we see with glufosinate. So again, the problem here is water hemp. And again, I'm sure all of you have seen many photos of water hemp or seen it in real life more than you care to admit to. This last photo is one my colleague, Dr. Aaron Hager, took of me. He incorrectly accused me of trying to repurpose water hemp as a form of a biological umbrella. That is completely false. I was just impressed with this impressive specimen of water hemp that I had collected. It is an impressive weed. I just want to highlight briefly some of the biological characteristics of this plant that make it such a challenge to manage for corn and soybean producers here in Illinois and in the Midwest. So one of its characteristics is that it doesn't just emerge in the spring and then it's done for the growing season. It has multiple flushes of emergence. It seems like every time we get a rainfall event, anywhere from about April into August even, we'll get a new flush of water hemp. And so if you're trying to, do an application early on, that's fine. That's going to control those early flushes, but it's not going to persist and control those late flushes. So it doesn't, this species is not conducive to just a single pass program. And if you're trying to wait till, okay, I know there's going to be some later flushes, I'm just going to delay my herbicide application. Well, by the time you get around to that last flush or those later flushes, now those water hemp plants that emerged early on, they are too big and they're past the window for good control. So that characteristic, this extended emergence through the growing season in and of itself can make it a challenging weed. And then when you do get escapes, if you don't get complete control, it doesn't take a lot of water hemp plants to really repopulate your field. That produces a large number of these very small seeds. All these seeds in these containers, there's 5 about 2-ounce containers filled with water hemp seed that came off of a single plant. You can get potentially 1 million seeds from one plant. So like I said, doesn't take too many to repopulate the field. A few escapes, you can go from a bad problem to a worse problem in just a year or two. And then the third biological characteristic I'll highlight is that water hemp is a dioecious species. So plants are either male or female, which means that it takes 2 of them to cross, and so they are always outcrossing. So there's a lot of genetic diversity in this species, and it's very good at moving through the pollen, wind-blown pollen, moving resistance traits from one population to another. And it's particularly good at stacking resistance traits together, just like we have crop cultivars now, hybrids that have stacked traits, stacked resistance traits. We have a lot of water hemp populations now with stacked herbicide resistance traits. So just kind of a historical perspective on water hemp's evolution of herbicide resistance. This is not a new problem that we've been dealing with. Essentially, as long as we've been fighting water hemp in our state, we've been fighting herbicide resistance in water hemp in this state. Beginning back in the 1990s with things like atrazine and the ALS inhibitors, progressing through most of our major post-emergent herbicide options we have in corn and soybeans for water hemp control. And now more recently with even a major group of herbicides, group 15, which is used for soil residual control of water hemp. So the VLCFA inhibitors or the chlorosetamide herbicides. So lots of resistances in that water hemp has evolved. But the real challenge is if I have a field with just one herbicide resistance or water hemp with just resistance to one herbicide, I'm not too worried about that. I can use other herbicides. But increasingly, what we're seeing is populations that have resistance to 2, 3, 4, even more, up to 6 different herbicide resistance groups within a single population, and sometimes even within a single plant. So literally getting to the point where we're about out of options for chemical control of water hemp in our state. And to put this in a little bit better perspective, I've listed here in this table all the different herbicide groups that are options for water hemp control in corn and soybeans in Illinois. And then the right-hand side, I've listed the resistance status. So a plus means there's resistance in water hemp to that group, and the more pluses there are means the more common it is that water hemp has that resistance. And so at the top there, you can see resistance to group 2, the ALS inhibitors, is very common in water hemp. It's hard to find a water hemp population in Illinois that doesn't have that resistance. And so you go down through the list of all these herbicide options, you see a lot of pluses here, right? In fact, the only place where the only 2 groups where you do not see a plus are the group 3, the dinitroanilins. We have no confirmed resistance to this group of herbicides in water hemp. The dinitroanilins would include the most common product that we might use here would be Prow or Prow H2O. It has no post foliar activity on water hemp. It's a pre-emergence product only. Very dependent on rainfall for effectiveness, so certainly not something we'd want to have to use as a standalone water hemp management tool. And then the other one would be glufosinate. I have a question mark there. We're going to talk more about that in detail today. But again, as you can see, we are literally running out of options or at the verge of running out of options for chemical control of water hemp. Before focusing in on glufosinate, the last trend I want to share with you in terms of resistance evolution in water hemp is depicted here. And so going back about 15 years, if we look at what are the mechanisms of resistance that we had in water hemp, most of them were target site mechanisms. Okay. And I'm not going to go into a big discussion about target site versus non-target site resistance today, but I just want to highlight that target site resistance is relatively easy to manage because target site resistance only affects herbicides that have the same target site. So if I have resistance to group 14 herbicides due to target site, I can manage that quite simply by rotating or mixing herbicides that have different group numbers, different target sites. Okay. Now, jump forward 15 years since 2010 or so. And what's really amazing is that all new cases or all new mechanisms of resistance that we've identified in water hemp in the last 15 years have been non-target site mechanisms. So things like situations where the water hemp has evolved the ability to more rapidly metabolize the herbicide. Okay. And from a practical standpoint, what this means is this non-target site resistance is harder to manage and it's less predictable because whereas the target site resistance only affects herbicides with the same target site, this non-target site resistance or metabolic resistance can affect herbicides that span across different target sites. So I could have a herbicide that selects for resistance to a group 2 herbicide. That resistance, that non-target site mechanism, might also confer resistance to a group 4 herbicide. Okay. So the point of this, from a practical standpoint, is that managing herbicide resistance is not as simple as rotating or mixing sites of action anymore. 15 years ago, that was our recommendation. We've had to change that recommendation now due to this increasing commonality of non-target site resistance mechanisms in water hemp. Okay, so that's a quick introduction to water hemp and herbicide resistance. So now I want to turn and talk about glufosinate. So glufosinate, as I'm sure most of you know, it's a contact herbicide that's non-selective. Obviously, it can be used selectively in our corn and soybeans that have been genetically engineered to survive this herbicide. It's a contact herbicide, which means that you have to have good foliar coverage for it to be effective. It's also, as a contact herbicide, it's not going to move much in the plant. It's not going to be as effective on larger plants. And so you want to spray plants when they're small, get good foliar coverage. I say slow acting, but just to clarify, it's faster acting than our systemic herbicides like glyphosate, for example. But compared to some of our other contact herbicides, like maybe the diphenyl ethers, it is a little bit slower acting. In addition to this, the contact herbicide characteristics making it a little bit harder to use effectively with the small plant size and the good coverage, it also tends to be a little bit more dependent on weather than many of our other herbicides. So it is kind of has a reputation of being kind of a finicky herbicide. So what are some of the things you need to do to be successful with this herbicide? And here's 3 bullet points, and these are just off of one of the glufosinate product labels. Spray when plants are small. Again, it's a contact herbicide. And it says in the label, less than 3 inches, right? That's pretty small, smaller than most people are actually probably spraying their weeds. And then again, you want that good coverage. So using medium coarse droplets, 15 GPA as a minimum, 20 gallons per acre is probably going to be better than 15 in most cases. So high volume, good coverage, small plants. And then beyond that, on the label, it states that control is generally going to be improved under conditions of high temperature, high humidity, and high light intensity. So what about resistance in to glufosinate? That's actually one of the advantages of this herbicide. It has very few cases of resistance compared to most of our other herbicides. In fact, worldwide, there's only 6 weed species that have been officially documented to be resistant to glufosinate. 5 of these are grass weeds, and unfortunately, one of them is Palmer amaranth, the weed that some of us also know all too well. And of course, Palmer amaranth is a cousin to water hemp. To date, there is not officially confirmed glufosinate-resistant water hemp that's been documented. But over the past couple of years, my colleagues like Dr. Hager, as well as other colleagues across the state and other states as well, have had multiple reports of less than satisfactory control of water hemp with glufosinate. Now, I talked about the fact that it can be a finicky herbicide. And so just because you don't get good control following an application doesn't necessarily mean you have a resistant population. Maybe that it was too cool. Maybe it wasn't humid. Maybe the plants were stressed for some other reason. And so we we tend to get, a fair number of reports of incomplete control with glufosinate, but a lot of times we can't rule out the environment, and so we haven't been able to confirm resistance. Although some of these cases have risen up, like I said, my colleagues, they will have conversations with farmers, maybe a field visit, try and troubleshoot, was there an environment that went wrong, or is it really a resistant population? And so over the course of the last couple of years, um, we've actually identified 4 populations across the state. This isn't an area that's unique to some corner of the state, but 4 different populations where there's reasonable suspicion of having some populations that are not being controlled effectively by glufosinate because of resistance. And so we've been investigating these populations. The way we typically do this is we take the seeds from the field site, collect plants, harvest that seed, and then we do what we call a dose response experiment. Simply grow up the plants in the greenhouse, grow a known sensitive population for comparison, and then treat with different rates of the herbicide and see, does the suspect resistant population respond differently than the sensitive one? So here's an example of one of those such experiments, showing here 2 different populations. That population from Northern Illinois, Carroll County, is on the left here, and then our standard sensitive population on the right. We treated with different rates of glufosinate in the form of Liberty Ultra. Just remind you that if you haven't used Liberty Ultra yet, typical use rate is probably going to be about 24 ounces. The maximum labeled use rate of Liberty Ultra is 29 ounces. And so if you look at these photos of these plants exposed to these different doses, you can see that as you increase the dose, you get better control. And by the time you get up here to a use rate, or actually just a little above a use rate, we have complete control of the sensitive population, but there's still some survivors in this Carroll population, even up to well above the labeled use rate. Some of these plants are surviving. So a clear difference. This looks like a clear case of resistance to glufosinate in this Carroll population. Having said that, though, I'll share with you that I've sort of cherry-picked these photos because we've done dose responses like this with this population, with those other 3 populations I showed you in that state map. And sometimes the results look very convincing, as they do here. Sometimes we see very little difference between the resistant and sensitive population. Okay. And so even in our, quote, controlled greenhouse trials, there's something that's going on here in the environment or something such that our resistant and sensitive populations are not responding consistently, and they're not consistently different. And so this really, begs the question of what's going on here. Why is this happening? Can we understand what the specific conditions are that affect this? Because if we know that, then maybe we can use that to our advantage to improve the consistency of control of water hemp overall, as well as to improve control of these suspect resistant populations. And so this really leads me into this ISA-funded research to try and understand this in more detail. And so more specifically, some of the questions we had were, we know light intensity, humidity, and temperature, time of day of application can influence glufosinate activity. So can we systematically isolate these different factors and see what effect they have, not only on water hemp in general, but also do they differentially affect sensitive and resistant populations of water hemp? Obviously, one of our goals is to optimize control of water hemp by finding those optimal conditions for glufosinate efficacy, but also we want to identify the conditions that will minimize the difference between the sensitive and the resistant populations, right? In other words, if we can identify a condition where the resistant population behaves like a sensitive population, then that is going to give us a way to potentially of controlling those resistant populations, but also a way of preventing the selection for resistance when you're treating populations that are largely sensitive populations. So try to control resistance that we may already have and prevent new resistance to glufosinate evolving in these populations. So those were our general questions. Our approach, again, was just to do the standard dose response experiment using this Carroll population and our standard sensitive population. But rather than just doing it in one environment, we grew the plants in a common environment, sprayed with glufosinate, and then transferred them to 5 different environments to understand and systematically tease apart the different components of those environmental conditions that might be giving those differential responses. Okay, so the way we achieved these for these environments was using growth chambers, which we could very precisely control temperature and humidity. And so we set up 4 growth chambers with every combination of high and low humidity and temperature. So high temperature, high humidity, low temperature, low humidity, high of one and low of the other. One of the downsides of using these growth chambers is they don't provide a real high light intensity. And so we know that light intensity is important as well. So we included a 5th environment, which was just a greenhouse room where we would have high light intensity. In the greenhouse room, we don't have as tight control over temperature and humidity, but generally the temperature and humidity was similar to our high temperature, low humidity growth chamber. So what do I mean by high temperature, high light, and high humidity? So the high temperature was not really hot. The low temperature was not really cold. We tried to use temperatures that you might encounter in June when this herbicide is being sprayed, but at the extreme. So sort of a hot day in June versus a cool day in June. And similar, that's how we pick the humidity conditions that we use. So representative of what you might experience in June, but at the extremes of what you might experience. And then in addition to those 5 different environments and having the 2 populations, we also had 2 different application timings. So glufosinate was sprayed either midday or in the evening. So if you do the math on this was a pretty intensive experiment. We had 2 populations. We had 8 doses of glufosinate. We had multiple plants per treatment. We had the 2 application timings. We had the 5 different environments. We repeated this experiment. So in total, we had 1,600 plants that were sprayed and evaluated under all these different conditions. And so a shout out to graduate student Isabel, who was able to pull all this off and collect all these data. Okay, so moving into the results. This is a very busy slide, a lot of data here. So be patient. Let me walk you through what you're looking at here. Okay. So on the y-axis, I have the LD50. So that is the concentration of glufosinate that was required to kill 50% of the plants. Okay, lethal dose 50. Okay, I apologize, that's in grams of AI per hectare. Don't worry about the dose. The absolute dose is not important for interpreting this. What you need is, the way to look at this is the taller the bar, the more glufosinate it took to have a similar effect. So tall bars are bad. Glufosinate's not working as well. Short bars, the glufosinate's working a lot better. You have 2 colors of bars. The blue refers to the Carroll population. The orange refers to the sensitive population. We have the 4 growth chambers, so combinations of temperature and humidity. So this will be the low temperature, low humidity, for example. Here's the high temperature, high humidity growth chamber. And this was the greenhouse room here. The other thing you see, so in each of these environments is 2 sets of bars. So one of those corresponds to midday application, the other to the evening application. Okay, so that's kind of what you're set up. So now let's walk through here and what can we interpret from this? What are the conclusions from this? So the first thing I want to mention is the midday versus evening. If you look across the growth chambers, comparing the sets of bars between midday and evening, you don't see a lot of differences and they're not consistent. And I think the reason for that was, as I said, the growth chambers do not have real high light intensity. And so I think the effective time of day is not as relevant when the light intensity is not as high. If you look in the greenhouse, though, where there is high light intensity, what you see is the midday applications for both the resistant and the sensitive population, you had better control. It took less herbicide to achieve that 50% lethality. So that's good. That's what we expected, right? Midday applications are better than evening applications. Okay, the next thing I want to highlight, well, just looking at comparing the colors, the blue versus the orange, the Carroll population versus sensitive. Very consistently, the Carroll population, those blue bars are taller than the orange bars. It took more herbicide to achieve the same lethality of that Carroll population as it did the sensitive population. So consistently across all these environments, the Carroll population is less sensitive to glufosinate. So I think, that's sort of one of the definitions of resistance, right? Where you have a differential response of 2 different populations of the same species. And so we see a clear difference there. Okay, the next thing I want to focus on is humidity. So looking at the 4 growth chambers, The two growth chambers on the right are high humidity. The two here are the low humidity. So going from left to right, it's same temperature, but increasing humidity. And what you see in both cases, holding temperature constant, but increasing humidity, greater humidity, the bars go down, the bars are smaller. Just what you think, right? What's the label says. Better efficacy if you spray at high humidity, and we see a clear illustration of that. So far, everything consistent with what we thought, no surprises. What about temperature? Let's look at the sensitive population. So now we go compare the two growth chambers above and below. So now humidity staying the same. You're going from low temperature to high temperature, low temperature to high temperature. First thing I'll point out about the sensitive population is it didn't matter quite as much what environment we used. We got pretty good control of that sensitive population. If you had high temperature, you got good control. If you had high light, you got good control. Where you see your control slipping is when both things are wrong. So you get low temperature and low humidity. Okay, low temperature and low humidity. Now we're not getting good control of that sensitive population, which again, that's what we expect based on the label. Higher temperature, higher humidity increases control. We saw that here. It looked like if you had either high temperature or high humidity, you got good control. What about temperature effect on the Caro population? Here was a surprise, at least to me. What we look at here is comparing, especially here looking at high humidity. If you go from high temperature to low temperature, control of the resistant population actually increases. In fact, at low temperature and high humidity, there's very little difference between the two populations. In fact, it wasn't statistically significant. So we're able to almost make a resistant population look like a sensitive population if it was sprayed and then the plants were in an environment of low temperature and high humidity. So another way to sort of look at those data is looking at the resistance ratio. So the resistance ratio is basically asking how much more herbicide did it take in the Caro population to get the same level of control in the sensitive population. And so 1 would mean that the Caro and the sensitive population responded the same. 3 up here would mean that it took 3 times as much herbicide on that resistant the Caro population to get the same control as you got on the sensitive population. And I said one of our objectives was to try to minimize this. What could we find an environment where the resistant and the sensitive population have a resistance ratio of 1? And we see that at that low temperature, high humidity condition, we're getting pretty darn close to 1. Low temperature and low humidity, you're not too far away from 1, but you're getting poor control of both populations. So we don't want that. High temperature, low humidity was the worst situation from a resistance ratio. Okay, so that's the one where you're really going to select that resistant population. And high temperature, high humidity is the best one for control of the sensitive population, but you're going to select for resistance. So there's a trade-off. Maybe by spraying at that cooler temperature, you're giving up a little bit of control over the sensitive population, but you're decreasing your selection for resistance. Okay, here's the same data set, but looking at it a different way. So here we're looking at the LD90. So what is the glufosinate rate that's required for 90% lethality, killing 90% of the plants? And so that's 371 grams of active ingredient per hectare is equivalent to that typical use rate of 24 ounces of Liberty Ultra. So all the blue symbols are the Caro population, all the orange are the sensitive population. You can see that most of the blue symbols lie to the right of that 1x rate. And so we're not getting 90% control of that Caro population under most conditions. In almost all the conditions, regardless of the conditions, we're getting, except for one there, we're getting better than 90% control. So high temperature, high humidity, which the label would tell you give you the best application or the best activity, that's true for a sensitive population, but not for the resistant population. The resistant population, if you spray or the plants are exposed to low temperature and high humidity, then we're actually getting greater than 90% control. And at those same conditions of the sensitive population, we're giving up a little bit of control, but not much. Okay, another way to summarize. So we're looking at different effects, right? We're looking at the effect of population, the effect of humidity, time of application, the effect of light, and the effect of temperature. Which of these were most important? How do they rank? Sort of averaging over everything else being equal. So the most important factor was population. Average across all the other conditions, the resistant population um, required about over twofold difference in terms of efficacy of glufosinate. You had to double your glufosinate concentration, all else being equal, all else averaged across all the other conditions to get the same control you got in the sensitive population. So that was the most important factor. Humidity was almost as important though, just under twofold difference. So having a high humidity improved that efficacy by about twofold. Time of application differences, as I mentioned, we didn't see probably as much effect as we thought we would, but I attribute that to the low light in the growth chambers. In the greenhouse, it was the third most important, causing a little bit less than the twofold difference. Highlight was we didn't control that as well. We had the greenhouse versus growth team versus the growth chamber comparison. And so that fell in there at being the fourth most important and causing 1.6-fold difference. And again, most interesting, I think, was the interaction between temperature and population. So temperature was the fifth most important factor, but there was an interaction such that with the sensitive population, high temperature increased efficacy by about 50-fold, whereas with the resistant population, high temperature actually decreased efficacy about 20-fold. So that's kind of a summary of these different effects of humidity, temperature, light, how that might affect your efficacy, all else or averaged across all the other conditions. So implications, first of all, I think there's a question, why is that resistant population, that Carroll population not being controlled as well at high temperature? And we don't know, I'll be honest with you, but my hypothesis is that at that higher temperature is allowing it to detoxify glufosinate faster. So we think it has a metabolism-based resistance mechanism, and we know that herbicide metabolism generally happens faster under higher temperature conditions. And so with that ability to metabolize that herbicide, it can do it faster at that higher temperature, and therefore it's less affected by the herbicide. From a practical standpoint, summarizing again, just restating this high temperature, we know it improves control in general. So if you have sensitive populations, but it might provide worse control of a resistant population and thereby increase selection for resistant plants in a sensitive population. So if you have a resistant population, you might think twice about spraying it when it's really hot. If you have a sensitive population where there might be a few individuals in that population that are beginning to evolve resistance, you might think twice about spraying it when it's really hot. I should admit, I'm saying this fully aware that you guys, when you're out spraying your field, particularly your custom applicators, you can't sit in your chair and wait till you get the perfect environmental day and then make a spray, right? When it's time to spray, you got to be spraying. If it's not too windy, you're probably going to be spraying. I understand that. So I realize the potential challenges in implementing what I'm saying, but nevertheless, I think it's good to be aware of how this temperature effect that we're seeing could be differentially affecting sensitive and resistant water hemp populations. So the ideal condition might be, for managing water, it might be that, spraying at midday when it's clear, when it's humid, and maybe when it's a little on the cool side. And I want to emphasize, I put a might in here. This is a greenhouse experiment. We need more work. I'm not ready to take this to the field. And so I don't want you leaving saying, hey, Professor Tranel said that we all need to spray glufosinate when it's cool. I think there's suggestions that needs to be looked at more, but we're not quite there yet. And I also want to point out some, additional sort of caveats of this experiment and some opportunities for future research. We looked at the conditions after we spray glyphosate. We didn't look at conditions before. How important is high temperature the day before you spray? We did not address that. I think that's something that needs to be addressed as well. We don't know. We have a better idea of some of the conditions that make for optimal efficacy, such as high humidity, maybe high temperature for a sensitive population, low temperature for the resistant population. But when should that occur? And for how long does that need to occur? So if it's cold the day you spray and the day after you spray, is that enough to improve efficacy of the resistant population? Or do you need 3 or 4 or 5 days of cool temperatures? We don't know that. So those are some additional unanswered questions. I also just say again, the growth chambers, the weakness is they had relatively low light intensity. And so how might high light interact with high temperature, low temperature, high humidity, low humidity? So there's some more combinations that we can do there. There's probably an infinite number of combinations. We can pick and choose which ones we need to do. But there's definitely some more that I think should be investigated. And I think this observation that the resistant population we had better control when temperatures were cool is really interesting, but we need further confirmation of that. Okay, so I want to wrap up here with a little bit more data. We've done some work regarding the mechanism of resistance. I want to share that with you briefly, as well as some work we did on site at that Carroll, where we collected that Carroll population. And so in terms of the resistance mechanism, I mentioned that Palmer amaranth has already been reported to be resistant to glufosinate. At least some of those populations have been shown to have increased copies of a gene encoding the target site, which happens to be glutamine synthetase. Similar mechanism to what we've seen for glyphosate resistance in Palmer amaranth and Waterhemp, in that they have increased numbers of the target site gene. The point is that it's a target site mechanism in Palmer amaranth. However, from our preliminary work, we saw no evidence of any changes in the target site in Waterhemp. So we don't think glufosinate resistance in Waterhemp is due to a target site mechanism. We've looked at differential gene expression in these populations, and we do see genes related to, associated with metabolic processes. So it's consistent with our greenhouse growth chamber study, and that we think that this resistance is due to metabolism. But again, we have a long way to go to confirm and verify that. So it does look, though, that glufosinate resistance in Waterhemp can be due to non-target site changes. So going back to what I said earlier on, what that means is rotating and mixing herbicides with different group numbers is not going to be sufficient for mitigating this resistance. The herbicides that are selecting for genes that can metabolize glufosinate, or there might be herbicides other than glufosinate that can select for genes and enzymes that can metabolize glufosinate, even though those herbicides are in a different group number. And then the last thing we learned about the mechanism, and so this is basically clustering the different populations based on their gene expression profiles. And without going into a lot of details here, what's interesting is that of the 4 resistant populations, 3 of them formed a unique cluster by themselves. The 2 sensitive populations clustered over here along with one of the other resistant populations. So what does this mean? Preliminary again, but it suggests that perhaps these 3 resistant populations here that are falling on this common branch, this common cluster, they might all have the same type of resistance mechanism. Whereas this population over here could have a different resistance mechanism. Okay, the Carroll population was one of these 3 here. And so point is that I provided some observations on one of these resistant populations, but there could be different resistance mechanisms out there to glufosinate. And so what you see, if you have a glufosinate resistant population, it might be different than what we saw with our population. But what this suggests, our sample size is only 4, so it's not a very powerful suggestion, but it suggests that most of these populations we think might behave like the Carroll population. But again, it's a very small sample size, very preliminary. Okay, the last thing I want to share with you is a field study that we did. We had this population. We had evidence that from Carroll County, we had evidence that it was resistant. We wanted to see, does it behave as a resistant population in the field? So this was kind of a last minute study. It wasn't put together quite as well as probably it should have been. But regardless, what we were able to do is make an application to glufosinate at the site where this population was collected. We actually we removed the crop, removed plants from the location so we could make sure we had water hemp plants that will be exposed to a full dose of the spray. When we made this treatment, when my students made this application, there was plentiful soil moisture. It was very hot. It was very humid. It was very sunny. It was an ideal day when you would expect to see absolute great control with glufosinate. We identified specific individual plants that were the appropriate height to spray. So here's the results. Again, here's this 371 would be a typical 1x rate. Looking at the percent control, just a visual observation of control, 6, 12, and 24 days. We got pretty good control. But again, conditions were about as perfect as you're going to get for water hemp control. But we didn't get perfect control. We had some plants that survived. And so, they were treated when they were small, conditions were ideal. 24 days later, they were still alive. So, not the control that you would expect given how good the conditions were. And so I shared this table with you before. I had a question mark with glufosinate. I think given the extensive greenhouse growth chamber studies that we've done, the consistent difference we see under those conditions, as well as the fact that under ideal conditions in the field, there were a few plants that survived. I think we can switch this to a plus now and say that that's another group of herbicides or another herbicide for which we now have resistance in at least some of our water hemp populations in the state of Illinois. So with that, I want to acknowledge my colleagues, Dr. Aaron Hager, as well as my students and others, colleagues here at the University of Illinois, as well as Carla Gage and her group at Southern Illinois. Her team and our teams here at Illinois have been working together on some of this work, trying to understand glufosinate resistance in water hemp and this variable responses of these populations. I also want to thank the Illinois Soybean Association for funding my work. And again, the opportunity to present this and for attention of all of you to sit through this. Leave this slide up with my contact information for a minute or so, and then be happy to jump into the questions.
Yes. Thank you, Dr. Tranel. First question I have for you. This person asked, what does the symptomology look like on a glufosinate resistant individual plant?
Good question. It looks the same as what the symptomology looks like on a glufosinate sensitive plant, except not as severe, I think would be the best way to describe it. So you're still going to see the symptomology. You're going to see that burning, but you're not going to see the continued progression of that. You're going to start seeing regrowth. So it just looks like, treat a sensitive plant with a reduced rate is kind of what you would see if you treat a resistant plant with a full rate.
Next question for you, Dr. Tranel. With water hemp's demonstrated capability to quickly develop resistance herbicide, how long do you project until chemical control becomes ineffective in the Midwest? I know you alluded to this earlier in your presentation, but do you want to dive a little bit deeper there?
Um, I honestly think we're getting to that point already. Chemical control is becoming less effective. I wouldn't say it's completely ineffective at this point, but it's definitely less effective. And I think we're to the point where we can, or at least we shouldn't be relying on just herbicides anymore. I liken it to like around 2010 when we had glufosinate resistance happening, right? We got to the point where we all realized that we couldn't rely on glyphosate. I meant glyphosate. When we started getting a lot of glyphosate resistance back around 2010, we recognized that we couldn't rely on glyphosate anymore as a standalone weed control tool. We still use a lot of glyphosate, but it's not a standalone tool. I think it's the same with our herbicides in general now. We're still going to use herbicides, but we shouldn't be using them as standalone tools. We need to be incorporating them with other non-chemical management strategies.
In the chat, one question we received. So when we blanket the countryside with glufosinate applications during periods of hot weather, are we likely selecting for metabolically resistant water hemp populations across a wide geography?
As this person, I'm not quite sure I understand the question. Obviously, the more we're spraying, the more plants that are exposed, the greater the chance that an individual plant that's able to survive that herbicide is going to be selected and allowed to reproduce. And I think that the key to delaying the evolution is to expose fewer plants to the herbicide. And when plants are exposed to the herbicide, don't let them set seed.
Another question from the Q&A, are certain adjuvants other than AMS which may help to improve control?
Yeah, I mean, the general recommendation if you're spraying straight glufosinate is to just use AMS. I know if you're tank mixing with some other products, there occasionally are some other adjuvants recommended. it's hard to evaluate every combination, and there may be some, what happens is someone might try an adjuvant and it might work pretty well, or it looks like that worked pretty well. Did the adjuvant actually improve it, or was it just they got one of those conditions where it was hot and humid, high light, and it seemed to work pretty good? And so I think the environment, I guess my long way of saying, I don't have any evidence that there's anything really better than AMS in terms of an adjuvant out there that's going to be consistently help out glufosinate.
Sure. Another question we got in the registration form. Do you expect water hemp to develop resistance to our commonly used group 14 and group 15 residual herbicides in addition to the resistance to post-emerge methods?
Yeah, I touched on that in that table I put up with the different herbicide options, and I had some pluses there for both the group 14 and group 15 herbicides. So we already have resistance to those products. Um, if I can take a couple minutes, I'll explain it. It's a little bit different. So when you have a
Yeah.
post-emergence herbicide and you have resistance to that, it's kind of black and white. You spray it and it doesn't die. Okay. Assuming it's not the environment that's resistant. With the pre-emergence product, you're spraying that before some of the weeds have even germinated yet. And so you're spraying that hoping that you're going to get maybe 4 weeks of residual control. But if you have resistance, particularly a low level of resistance, the way that might manifest itself is instead of getting 4 weeks of control, you may only get 2 weeks of control. So those pre-products, they're still giving you value, it's giving us value on most of our acres, but because of resistance to them, they're not as effective as they once were. So that resistance already is out there.
Another question we got, and if you have any more questions, feel free to use the Q&A chat feature. One more question. What do we do once Liberty no longer works?
That's that's the question, right?
That's the golden question.
Yeah, I think the mindset among a lot of farmers is that companies have another product that's out there and is going to be ready to go. And certainly they are working on new products, and there's hope and there might be some new products. But even if they come out, commercialize a new product, it's not going to be a cure-all. There's going to be limitations. You know, think back if you were farming back in the day when glyphosate was working really well. If you could go back in time, would you use glyphosate the same way you did? I hope some of you would say no. Hope some of you would say no. We would try to rotate it, mix it, continue using pre-emergence products. If we would have done that and treated glyphosate as a tool, not as a tool, not the tool, we might still be using glyphosate today. So I think, whether it's glyphosate, glufosinate, some new herbicide, we have to treat it as one of many tools rather than just rely on it. Because, as I showed you in that table, we don't have, once we get resistance to glufosinate and that becomes widespread, we get widespread resistance to 2,4-D, there's really nothing left that we have post-emergent to control water hemp in our state.
And okay, we just got another question from our Q&A. Late season water hemp suppression, and that's in quotes, applications of glufosinate do take place even off-label. What role is that playing in selecting for resistance?
Yeah, that's a good question. Obviously, when you are spraying a plant with glufosinate and not killing it, that's a bad thing from a resistance select standpoint, right? Because you kill the wimpiest ones, right? And so the big ones, so maybe the big ones are completely sensitive, you'll kill, but if you have a big one that has the ability to metabolize the herbicide a little bit faster, and the fact that it's big allows it to survive, now it passes on those genes to the next generation. And so that's not a good thing. And so, like I said, you want to limit how many plants you get exposed to glufosinate. And you can do that by using pre-emergence products, using cover crops. And then when you do spray plants with glufosinate, you want the ones that are exposed not to survive. And you can do that by mixing it with another herbicide or following it up sequentially with another herbicide or going out there by hand and getting rid of it. Because if a plant is exposed to glufosinate and it survives and it sets seed, that's how resistance can happen. If it's not setting seed, then it can't, resistance can't evolve.
One clarification question we just received. What percentage of humidity is considered high?
Yeah, so I had that in my note. I think 70% is kind of thought of as a cutoff. Above 70% would be where you see pretty good control, you get below that 70% and it starts dropping off.
And then a follow-up, what temperature is considered low?
Yeah, so in our experiment, our low temperature, I believe, was 70 degrees that day and 60 at night. Again, that would be a cool day in June, but not completely uncommon day in June. You know, would you see the same thing at 75, 80, 65? We looked at two temperatures, and so haven't looked at a complete range. And is it kind of just a gradual thing as you increase temperature? Or is there sort of a threshold temperature? We don't have the answer to that at this point.
Another question from our Q&A. Do you think conditions before application or after application are more impactful for a successful glufosinate application?
That's a good question too. I think it varies. I mean, humidity, I think the day of application is when humidity is most important. Um, Rainfall before application, actually, there's been some previous work by some of my colleagues here at the University of Illinois showing that rainfall, like 5 days during the 5 days before application, are pretty important because that, if there's plentiful rainfall, the plants are not stressed, they're actively growing. And then high temperature after. So humidity, I would say day of. Temperature, I would say after. Light intensity, probably the day of. But then rainfall would be before, so the plants are not moisture stressed. And all of those can come into play and interact.
Another question, does spray solution pH impact glufosinate efficiency or efficacy?
Yeah, I'm going to pass on that question. I don't
Okay.
have direct knowledge to answer that one.
Okay, that's okay. Another question, why are there not more efforts to develop non-chemical means of control to aid in combating resistant weeds and maybe to add on that lack of adoption as well?
Yeah, I think adoption is probably the key. I mean, I would say there is a lot of effort. There's work on things like lasers to weed, right? Robotic weeding, genetic strategies, using sterile pollen. There's a lot of research being done. Um, a lot of research on maximizing cover crops, how to maximize cover crop for weed control. Lots of research there. So I think the challenge is that it's more adoption, and when herbicides do work, it's really hard to compete with them in terms of speed, cost effectiveness, and so on. Necessity is the mother of invention, and I think we're starting to see that. And I think necessity is the mother of adoption. When a farmer doesn't have a herbicide option that's working post-emergence, that's when they're going to start using these other strategies.
One more question, what is more important, environmental conditions or size of the plant?
Um, it's hard to say. I mean, what size are we talking? If it's 3 inches versus 4 inches, it's probably environmental conditions. But if you're talking a 3 inch plant versus a 15-inch plant, then it's probably plant size that's more important. So
I believe that's all the questions we have. Dr. Tranel, after answering those questions, anything you want to leave our viewers with?
well, I think that's it. Those are some good questions, and yeah, appreciate your attention again.
Thank you. And Dr. Tranel, you can find his contact information. I know he showed it earlier. You could probably also research his contact information on the University of Illinois website. So, Dr. Tranel, again, thank you for coming today and discussing this. I know we wanted to get you to present at Forum on the 15th, but you were unable to come. So we thought, well, let's do a webinar on it. So thank you for taking time today.
My
And
pleasure.
also, thank you to the attendees for coming today and asking great questions. Remember, scan the QR code for CCA credits. And if you're watching the recording, you can email us at fieldadvisor@ilsoy.org for assistance to self-report. Lauren, if you want to go to the next slide. Speaking of forum, January 15th, 2026, at the Atkins Golf Club at the University of Illinois, we will have presentations on weed management, sulfur applications, nutrient management, um, economics, late season soybean management. So I guess what I'm getting at, if you enjoyed this webinar today, we will have a lot more information like this at our Field Advisor Forum on January 15th. So you can scan that QR code to register, or you can go to fieldadvisor.org. And if you just scroll a little bit, right there are events on the homepage, you'll find it there and the full agenda as well. And that event's brought to you by the Illinois Soybean Checkoff program. January webinar, we are working on the details for that. And we will have information very soon. So if you're on our eBlast e-subscriber list, we will get those details to you within the next week or so. And then also you can subscribe to Field Advisor via our newsletter, which you can sign up for on our website, YouTube, podcast, Facebook, X, Instagram. We're on all of those platforms. So thank you again for tuning in today, and we hope to see you soon, whether it's at Forum or our next webinar.